Alrighty, we are on page 23. I know we've finished this, but we're going to take another moment to go over it. Because at the end of last period, we learned a couple of new words, and we can actually put them in here. So in this diagram on the middle of the page, on page 23, I'll give you guys a minute to get that out if you don't already. We have DNA, and in the nucleus, we make a partial copy of the DNA, which is the RNA. Now, tell your partner if you can remember the name of that process by which we make an RNA copy of the DNA. Yes, that is transcription. So DNA to RNA, that is called transcription. That is a vocab word. And that takes place in the what part of the cell? Nucleus. We also have this other process here where we take RNA and we use it to make a protein. Tell your partner if you can remember what the name of that process is. That would be translation, yes. And that's when we read the RNA and we make a protein. In what part of the cell does translation occur? Does it occur in the nucleus? No, it occurs in the cytoplasm. And even more specifically, in the cytoplasm, it occurs in which organelle? It occurs in the ribosome. This specific type of RNA, this little partial copy of the DNA, has a special name. Tell your partner if you can remember the special name for that particular type of RNA. Ooh, mRNA. And the M in mRNA stands for something. Messenger. Okay. Yes. Oh yeah, this part here. There we go. Yeah, no problem. Yes. I think just because it makes the RNA like part stand out from the rest of it. That would be my guess. Yeah. Yes, Kanisha makes a good point. If you're writing mRNA, the M is lowercase and the RNA is capitalized. Okay. Okay. Now let's see if we missed anything on this page last day. We mostly managed to finish it. Do you remember what the word synthesis means? Yes. It means making. So in the nucleus, we make the mRNA. That mRNA partial copy then moves out of the nucleus. What does it go out of the nucleus through? It goes through a nuclear pore, which is a hole in the what? Nuclear membrane. And then the mRNA goes into the mouth of a ribosome, which is actually made up of two smaller parts. Do you remember what we call those two parts? You're very close, not big, but large. Yeah. Thank you. We have the large subunit. And we have the small subunit. All right, and it reads that mRNA and it assembles these amino acids together into a, and this is a black diamond word, a polypeptide is just an unfolded protein. It's a protein that hasn't folded up yet. But if you just wanted to say that it's making a protein, that's fine. You don't have to use that word polypeptide. The protein is what we call it after it has folded up. Do you remember what the prefix poly means? Poly means multiple or many. I'm going to use many because it's a short word and I can fit it in. Okay, so poly means many. And peptide means having to do with proteins. The bonds that form between amino acids are called peptide bonds. You don't need to know that this year, but just letting you know that's where the peptide part comes from. And there is an enzyme in your stomach that chews up proteins. So if you were to eat a hamburger and you have all that protein in there, the enzyme in your stomach that chops up the protein is called pepsin. So peptide has to do with proteins. All right. Good, good. We are now going to watch this video. We're not going to take notes or anything. We're just going to watch it and absorb what we can, and then we're going to go over it in slow motion, okay, with me. So let's just sit back, relax, and enjoy this video. Oh.
Today's video is all about DNA. So we're going to run through the structure of DNA with a focus on nucleotides and complementary base pairing.
Did you read the word nucleotide in your reading? Yes? Okay, if you didn't do the reading, nucleotide is going to be a vocabulary word. Did you read about complementary base pairing, or is that something new? You did read about it? Okay, if you didn't, that's okay, don't worry. It will come about. Again, don't worry, you don't have to. This is, what page are we on? It doesn't even matter what page we're on because we're just going to watch the video and then we'll go over it, okay?
And at the end, we're going to briefly go through how a gene can code for a protein. The first thing to understand about DNA is that it's made from two strands that are wrapped around each other in this twisting shape that we call a...
What do we call it? That twisting ladder shape is called a double helix.
Double helix.
Yes.
To understand the rest of the structure though, let's take just one of these strands and then show it as a 2D diagram, as though it's been untwisted and laid out flat. So we're now looking at this section here of the original strand. Now, hopefully, you can see that DNA is actually a polymer.
It's a what? A polymer. What does poly mean? Many. And what do you see here? We have many little subunits stuck together, yes? Yes, sorry.
Oh, that means like a hypothetical that monomer exists?
Yes, a single unit of this is a monomer, right? But when we stick together 1, 2, 3, 4, etc., then we get a manymer, a polymer. So one unit is a monomer, many units is a polymer, mer being the unit.
Because it's made up of lots of these tiny little units called monomers. With DNA, we call each monomer a nucleotide.
Okay, so what we're doing is we're taking this little bit here and we're blowing it up so you can see the nucleotide.
And if we look at the nucleotide in more detail, we can see each one is actually made up of 3 different parts.
See if you can remember from your reading what those 3 different parts are. It's okay if you can't, but see if you can remind your partner what those 3 different parts are.
Yeah, that sounds like you.
We got a P. Anyone remember what the P stands for? Yes. That is a phosphate. You remember PO4 from our chemistry unit? That's what's right there. That's a PO4. That's a phosphate. Anyone know what our little pentagon is? Yes. That is a sugar. In this case, because it's DNA, it's actually deoxyribose sugar. That's where it gets the deoxyribo part of its name. And then finally, we have this part here. Anyone remember what that's called? Maya. That is the base. Yes. So we have a phosphate, we have a sugar, and we have a base.
At the top, we've got a phosphate, which is connected to a sugar, and then on the side, we have a base. Importantly, every nucleotide has exactly the same phosphate and sugar. But when it comes to bases, there are 4 different types, namely A, T, C, or G, which stand for.
Okay, A is adenine. What is T? T is thymine. Remember, do you need to know these names or can you just call them A, T, G, and C? A, T, G, and C is good. But if you're interested, T is thymine, C is cytosine, and G is guanine.
Adenine, thymine, cytosine, and guanine. And this means that there are effectively 4 different nucleotides in DNA, one type for each of the 4 different bases. If you take a look at these 2 nucleotides here, we can see how they'd combine together to form a polymer. Basically, the phosphate.
So what we have here, we have a single unit. What do we call a single unit?
Monomor.
We call one unit A.
Oh my God.
What does one mean? What prefix do we use for one? Mono. So it is a monomer. Okay, so this is a monomer. This is another monomer. If we stick them together, what are we going to get? A polymer.
Phosphate of one nucleotide bonds to the sugar of the next nucleotide.
So we get a bond.
And this process then keeps repeating for thousands of nucleotides, so that the sugars and phosphates form one long chain, which we call a sugar-phosphate backbone.
Actually, I'm going to keep that going.
And if we look back at our full DNA molecule over on the left, the sugar-phosphate backbone is this outside part.
Okay, so this sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, that is this gray bit along the sides. It's the sides of the ladder, which are the sugar and the phosphate. So the sides of the ladder are identical. Okay, there's nothing different about them. It's only the bases that change.
So it's effectively forming a protective outer casing around those bases in the middle. If we look back at our single long chain though, you can see that all the bases stick out to the side. And these are what hold the 2 strands in the double helix together. If we line up a second strand of DNA, facing the opposite way, you can see how the bases could pair up and hold the 2 strands together.
So they pair up, they don't actually form a bond, they form a weaker structure. It is actually called a bond, it's called a hydrogen bond, but it's actually not a true bond. It's a somewhat weaker force. And the nice thing about that weaker force is if it was too strong, you could never separate the 2 sides of the DNA. And it's going to become important that we can pull those 2 sides apart. Because you can't make a copy of something if you can't get at the letters. So we need to be able to pull apart the 2 sides. So we don't want these bonds to be too strong. But what if they were too weak? What would happen to my DNA molecule if those little hydrogen bonds in between were too weak? What would happen? Yes? Yeah, it would fall apart and then you wouldn't have your 2 strands. And remember, it's really nice to have those 2 strands because then your code is protected by the outside sugar phosphate backbone. Yes, you want them to be together so that those bases are protected, but you want them to be able to come apart when needed. So that's why it's important that we have these hydrogen bonds in between.
Importantly though, only complementary bases can pair to each other.
Who are the complementary bases? Tell your partner.
C.
What? Who are the complementary bases? What that means is who pairs up with whom.
So A always has to pair with T, and C always has to pair with G. We call this concept complementary base pairing, and it allows us to figure out what the complementary sequence of a strand will be. For example, if we have a strand of DNA that reads A, G, T, G, C, T, T, A, C.
We are going to turn, if you are not there yet, to page 35. Page 35. Because we will see this little picture on page 35. Oh, and look at this. There's a nice little mnemonic device here on page 34. Where do the apples go? Apples go in the tree, and A pairs with T. Where does your car go? The car goes in the garage, so C pairs with G. How nice is that? Where does the apple go? In the tree. Where's the car go? In the garage. So that tells you which bases are. What's that word that starts with C that means they pair up nicely? Tell your partner. The word that starts with C that means they pair up perfectly.
It's like when we say two people on a date, you know, all the animals.
The word is complementary.
Okay.
Complementary, you did. Yes, complementary. That means they pair up perfectly. All right, page 35. Could you please use the apples goes in the tree and the car goes in the garage to predict what the complementary strand will read? So what you're going to do is you're going to write in whatever's going to go there, and you're going to go along and fill in the blanks with the correct complementary bases. Yes. Yes. Okay, who've we got here? We've got A goes with T. Everybody, what comes next? 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 Beautiful. That's guaranteed to be one of your questions on a test, is drawing a complementary set of bases. Is that easy? Hopefully, the apple goes with the tree and the car goes in the garage. Yes? You're good. Okay, cool. All right, back to our video.
Then we can use this sequence to work out what the sequence of bases on the complementary strand must be. Because we know that A always pairs to T, and G always pairs to C. So we know that the first base on our complementary strand must be a T, because that's complementary to the A. Then the second base must be a C, because that's complementary to G. Then the third must be an A, then C, then G, and so on. As a last point, whenever you hear the term genetic code, It's the sequence of bases that they're talking about.
So what the heck is a genetic code? It is literally just whatever the letters say. So it's the series of bases. So this genetic code reads AGTGCTTAC. That is the genetic code. Okay? So it doesn't mean anything fancy. It just means what do the letters say? That is your genetic code. Okay?
And a gene is just a particular sequence of bases that codes for a particular protein. To do this, each group of 3 bases.
So, what is a gene? It's a particular sequence of bases. So if I have a DNA molecule that goes from that wall to that wall, maybe this section right here is one gene. And if I read that code from here to here, 3 letters at a time, it will tell me what amino acids to put together to make one particular protein. So a gene, one gene makes one protein. Okay? When you get to biology 12 or university, you'll learn that genes are more complicated than that, but that's okay. That's all we're going to learn about it this year. Okay, there's all sorts of weird stuff happens with DNA when you get more complicated.
Is called a triplet and codes for a specific amino acid. For example, AGT would code for one amino acid, while GCT might code for a different one, and TAC would code for a third. To understand how this helps us code for a protein, let's take a longer sequence of bases and work through the steps for how it would form a protein.
Could you tell me the first set of letters that would tell us which amino acid? ATC. What comes next? GCT. Are you guys following? Okay. So we're going to watch them go through this.
First, our cells would read this DNA base sequence as a series of triplet codes, which remember are 3 bases each. Then it would take the amino acids that each triplet codes for and combine them all in that same order. Then lastly, this long chain of amino acids that we formed will fold up all by itself and form a protein. Now, the important thing about proteins is that because each type is made from a different sequence of amino acids, each type will have a unique shape, which allows it to carry out a particular function. So within each of our cells, we have loads of different proteins that carry out loads of different things. The main uses of proteins, though, are in enzymes, which act as biological catalysts to speed up the rate of chemical reactions.
Okay, so a catalyst is something that speeds up a chemical reaction. So in your body, there are enzymes, which are biological catalysts. And they usually speed things up, like you can have digestive enzymes. In theory, the food that goes into your stomach could just eventually fall apart, but that's going to take hundreds and thousands of years. Wouldn't it be nice if there was something to chop it up? That's an enzyme. It makes that breaking up of the protein or whatever food you've eaten faster, right? So you don't have to wait with the hamburger in your stomach for thousands of years to digest it. Those enzymes will speed that up so it will digest in a couple of hours rather than a couple of centuries or millennia.
Hormones, which carry messages around the body.
Hormones are like signals. What happens is they go into your blood and they just travel all around your body. So if your brain needs to get a signal that's not a nerve signal, if it needs to get a signal to the rest of your body to start making something or doing something, it can send out a hormone, which could be a protein.
and structural proteins, which add strength to our cells and tissues. Anyways, that's everything for this video. So hope that made some sense and helped, and we'll see you again soon.
Okay, so what we're going to do right this second is we're going to go here, and we're going to go back to the very beginning on page, what page are we on? And just make sure we've hit all our vocab words and explain anything that you missed, okay? So this is page 29, page 29. Now you should know that DNA, its shape is called a double helix. And if you had to explain that in terms that weren't so sciencey, what is a double helix? It is a twisted ladder, yes. So double helix is a vocab word. Okay. DNA is a polymer. Do you think polymer is a vocab word? Yes. Poly means many, and mer means units. So we have many units. How do you get a polymer? How do you get a whole bunch of units? Well, you need to take many of the single units, which is a monomer. So you have one unit. Do you think that's a vocab word? Absolutely. So a monomer is a single unit in a longer chain of molecules. So what's a monomer? A monomer is a single unit that we can put together in a longer chain. What is a polymer? It is that chain of repeating simple molecules, and those simple molecules are the monomers. So here we go over here, this whole thing. What would we call this whole thing? I mean, it's half of a DNA strand, but we would call it the whole thing, a polymer. So the whole thing is called a polymer, and a single unit of that is called a monomer. So if you bond a whole bunch of those monomers together, you make a polymer. Okay. Let me know if I'm moving too quickly. If I've moved it, we're good? Okay. So what is the monomer in DNA? A DNA monomer is called a nucleotide. So a DNA monomer is called a nucleotide. Do you think nucleotide is a vocab word? It sure is. Nucleotide. Now, if you're like, I suck at spelling, I'm never going to be able to remember the word nucleotide. Yes, you will, because what are you going to be making? A cheat sheet. Yeah. So if you're like, oh my gosh, so many vocabulary words with so many letters in them, it's okay. You're making a cheat sheet. Okay. So here is our DNA strand, which is the polymer. And then we have our nucleotide, which is the monomer. So if we string a bunch of these monomers together, we get a polymer, the DNA strand. And now we're going to zoom in on the nucleotide. And I'm going to get you guys to tell me what this P stands for. That stands for phosphate. Now, what shape is this thing? That is a pentagon. What does the pentagon represent? It represents the sugar. And the reason it represents the sugar is that literally the sugar is made up of 5 carbons bonded together in a ring that makes the shape of a pentagon. Okay? So that's why we have a pentagon. And then finally, what's this green bit over on the right? That's the base. Now, for those of you who want the sugar, what is the name of that particular sugar in DNA? Something that starts with a D. The sugar is D-oxy ribose. Now, do you guys know the name of any other sugars? Glucose. Any other sugars? There's glucose. Table sugar is... We have fructose. Fructose is fruit sugar. Do you guys know what milk sugar is? Lactose. So if you're lactose intolerant, you cannot properly digest the milk sugar. And the table sugar, the stuff that you would just have in, like, you know, you would sprinkle on your cereal in the morning or put in your coffee, that would be sucrose. What do you notice about all those names? They all end in ose. Ose is the name ending for sugars. And again, do you need to know that? No. So we have phosphate, we have deoxyribose sugar, and we have a base. Now, for those of you who are not doing enriched, we are going to specify the type of sugar here, but we're not going to call it anything fancy. We're just going to call it D sugar. That way you don't have to remember deoxyribose sugar. You can just be like D sugar, and it's DNA, so it's D sugar. So you should know D sugar. You should know the phosphate. You should know the base. And when we talk about this as vocabulary, it's like you should be able to say, oh yeah, I could recognize on a diagram where those are in the entire nucleotide. Now, what I'd like you to do is I would like you, and then we're going to take a little break, okay? I would like you to get some sort of piece of paper. I would like you to shut your book so you cannot see this lovely picture of a nucleotide, and I would like you to see if you can draw it from memory. Can you draw and label a nucleotide from memory? Once you show me that picture, you can take a 5-minute break. See if you can draw a nucleotide without looking at the picture. And if you have trouble, check with your neighbors. And if your neighbors have trouble, walk to someone else who isn't having trouble. We are drawing a nucleotide, and labeling the 3 parts. Don't look unless you have to. When you're done, you can show me, you can take your break. Beautiful. Nice. Nice. Good stuff. I want those to be attached to each other by bonds. Yes? That looks great. Can you label them? That looks good. That looks good.
I don't know how to spell frostbite.
That's okay. It is beautiful. Beautiful.
Thank you.
Oh, hang on. The phosphate is going to be the circle. The D sugar is going to be a pentagon.
Exactly.
Yes?
Yeah. Okay, thank you.
Beautiful. That's okay. A house is a good pentagon. Go for it. That looks great. Okay. That looks very nice. Yeah.
I accidentally drew a hexagon.
That's okay. It's all good. Thank you. Perfect. Excellent. Wonderful. Nice. Thank you. Nice. Nice. Thank you. Thank you. Hello. That looks good. Thank you. Excellent. Yes.
What is the answer to question 7?
7, your retina cells and muscle cells contain different identical DNA. They are able to function differently because they turn on different genes.
Okay.
Yeah. So there's certain genes where it's like, okay, make this protein.
Yeah.
There will be like chemical signals that come in. That is beautiful. You just want to make sure you attach those two with a line. Perfect.
Okay, thank you.
I'm also going to take my break because I've been drinking tea, so I might need to use the washroom. How's that? That's good.
Yeah, but I have to do the closer. You also have to. I think. You looked at the house. Yeah. No, I'm because you're not.
Alrighty, phones away, please. Now, one thing I noticed about people's diagrams is that the ability to make a pentagon varies wildly within the class. I saw octagons, I saw hexagons. So we're going to do a little practice just drawing some pentagons. How many sides on a pentagon? 5. So what we're going to do, you'll notice that, and if you're really good with shapes, you can take this into account. This one is actually slightly tilted. We're not going to draw them tilted like that because that's more complicated. If you want to do that, that's fine. Like if you've got that artist's eye and can remember it, that's great. But if you can't, let's practice just drawing some straight up and down pentagons. So what we're going to do is we're going to draw 2 lines like that. Okay? So it's like you're drawing the roof of a house. Okay? Once you've done that, you're going to draw a line that tilts. It's almost like you're drawing the sides of the house, except for you make it go inward just a little bit. And then you just have to connect the bottom. So once again, we're going to draw the roof of the house. We're going to make the sides of the house slope slightly inward. And then we're going to connect the bottom. Draw the roof of the house. Make the sides of the house slope slightly inward. And then connect the bottom. Please draw 5 more pentagons, just for practice. We've just been practicing drawing pentagons. For those of you who just came in, make sure that you draw the roof of the house like that. you're going to make this, it's like we have the sides of the house, but they tilt slightly inward. And then we connect the bottom. And we're drawing 8 pentagons. So we draw the roof of the house. We make the sides of the house go slightly inward. And then we connect the bottom. Draw the roof of the house. Make the sides go slightly inward. And then connect the bottom. Your job, should you choose to accept it, is to draw 8 pentagons, because I had all sorts of things that were not pentagons. There were octagons, there were hexagons, there were all sorts of things. So what we do to draw a pentagon is we draw like a roof of a house, essentially. And then you make the walls of the house slope slightly inward. And then we connect the bottom. We draw the roof of the house. We make the sides of the house so slope slightly inward. And then we connect the bottom. Okie dokie. I know it's hard. Draw the roof of the house. Make the sides of the house slope slightly inward. And then attach the bottom. Now, I'm going to show you how to attach the phosphate and the base. Okay, so we'll do this at the top of the next page. We've got, I'm going to put my pentagon right here. 1, 2, so sort of in between there. Oh, that's a bad place to put it. There's not enough room. Never mind. Let's do it over here. Pentagon. What do you think I'm going to attach to the top of the pentagon? Why am I labeling it S? That should be labeled P for phosphate. I can label this S for sugar. And then coming off the side, I draw a line. And then you can draw any sort of rectangular shape that you want here, okay? You can even just make it a rectangle, and that is your base. So you put a line off the top, you put a line off the side, and then you attach the phosphate and you attach the base. Is good? Cool. Now, for nucleotides, what do you notice about all the phosphates and the sugars? They're all the same. Nothing different. However, what about the bases? How many different bases are there? 4 different bases, and those bases are, tell your partner, either the letters or the actual names. Yes. Pardon me? Yeah. 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 Exactly. Yeah. And it's good you remember it's called the night project. comes from when you have nitrogen in the organic compound carbon-based compound, that's an amine. Ione. So that's what the ione comes from. Alrighty. So let's watch how these nucleotides combine to form a polymer. So what we're going to do is we're going to just attach one monomer to another monomer. We get a bond. And we keep attaching them and we keep attaching them and we keep attaching them until we get one long chain. So on the side, what do we call that chain of sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate? Sugar phosphate, sugar phosphate, sugar phosphate is called the sugar phosphate. Isn't that amazing? The sugar phosphate, sugar phosphate, sugar phosphate is called the sugar phosphate backbone. And that makes up the sides of the DNA ladder. And that is a vocab word. Okay. So that is our sugar phosphate backbone. It makes up the sides of the DNA ladder. Can I scroll or we need another minute? There we go. So what we're going to do here at the bottom of the page, if we have room, we are going to draw over here a pentagon, a smallish one. What will we attach to the top of the pentagon? A phosphate. What shape do we use for the phosphate?
Circle.
Circle. What do we attach to the other side? We attach a base. I'm just going to make it a rectangle. That's fine. So we got our base. Now, attached to the sugar at the bottom, what's it going to attach to? A sugar, yes. Pardon me? RNA has the sugar phosphate backbone and it has the bases sticking out the sides, but it doesn't pair up with another base. Yeah? So it still has the bases sticking out the side, but it's only got one rung of the ladder, like half a rung, rather than going all across the diagonal. What's the difference? It is very much the same. RNA and DNA are very similar molecules. Later on, I do have some pictures showing you the difference between the ribose sugar and the deoxyribose sugars. Not that you ever have to know that, but it's kind of nice to see. Okay, so I'll look and see if I can find those in just a minute. Okay. All right, now what are we going to attach to the bottom of this sugar? We're going to attach a phosphate. And then what goes below the phosphate? A sugar. So we're going to draw a line and we're going to draw a sugar. And then what's going to come off the side? A base. Okay, and you can put a P in the circle and an S in the pentagon. And there is your phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate. Okay, so here's our sugar phosphate backbone. If you want to, this part, you can highlight it. If you have a highlighter, that is the sugar phosphate backbone. And you know what it does? It protects the genetic code. It forms a nice shield along the outside that protects the bases. Because you want to keep your genetic code intact, right? You don't want it getting like knocked about and little repairs being made and oh, someone fixed it wrong and now it's a different code than it was before. That would be bad. So we've got that lovely backbone protecting everything. Okay. I feel like we've done a lot today. I would like you to, well, we got 20 minutes, so maybe you won't have homework, right? So, how many people completely finished pages 5 to 10? Okay, if you finish pages 5 to 10, you don't need to do it. But for those of you who didn't, right now you have an opportunity to work on that. Pages 5 to 10. And the second thing we're going to do is, shush. Pages 25 through 27. No, 28. This is last day's homework. So if you get behind, you're just going to keep having homework piled on top of homework, piled on top of homework, and that would suck. So don't do it. And also, if you can collect vocab words and diagrams. Now, we still have 20 minutes left. No, we don't. Because I thought this was block 4. What a silly person I am. Never mind. We have no minutes left. I thought this was block 4. Wow.